The Ethiopian Educational Media Agency presents educational satellite television programs.
Once again, hello teacher and students. I hope you enjoyed the first lesson, which, as you remember, introduced you to cells and the microscope. By now, you should have an idea of what a cell is, and you should also be able to explain how a microscope works. In today's lesson, we'll see how to set up a microscope, and we'll make two slides, one of a plant cell and one of an animal cell, so that we can view them under the microscope. Let's start by setting up the microscope. Firstly, remember to carry the microscope by the arm to prevent it from falling when you're moving it around. Place the microscope near the edge, but not on the edge of your laboratory table with the arm facing you. It will be easier for you to see through the eyepiece if you adjust your laboratory stool to suit your height. This part is called the condenser. What that does is concentrate the light so that it falls directly onto the slide. In some microscopes, the condenser can be lowered or raised. In others, it is in a fixed position. Now, raise the condenser closest to the stage. It will go when lowered. If your microscope has a microscope bulb, switch it on. If the microscope you are using does not have a built-in light, then switch on your table lamp, or you can place your microscope near a window. Now, look through the eyepiece and adjust the mirror until your field of vision is evenly illuminated. By this, I mean the area you are looking at must have enough light. Remember, once you have adjusted the light, you shouldn't move the microscope or the lamp. If you move them, you may need to readjust the light. You can use daylight by adjusting the mirror towards the window. Now that we have set up our microscopes, we are ready to study a mounted specimen or slide. The first step is to place the mounted slide on the stage with stage clips. Remember that the stage is the flat part here, and these are the stage clips. Next, you move the slide with your thumb and finger until the object is at the center of the stage opening and on the path of the light becoming from below. The stage clips will prevent the slide from moving out of the field of vision. If the low power objective is not in position directly above the stage opening, you need to hold any two objectives between your thumb and the forefinger and turn the revolving nosepiece until the objective clicks into position. Remember, this is the nosepiece, and these are the objectives. Notice that the shorter objective or the low power objective is labeled 4 times. The middle power objective has a magnification of 10 times. And the high power objective has a magnification of 40 times. In some cases, your microscope may have an oil immersion objective, which generally has a magnification of 100 times. Now, while looking at the microscope from the side, raise the stage with the coarse adjustment to within 5 millimeters of the slide. Remember that the coarse adjustment screw, which is this part bring about large movements of the stage, or in some cases, the body tube. It allows the objectives to move closer or further away from the object you wish to study. It is important that you know the functions of the various parts of the microscope. So, I will give you a moment to write down the function of an adjustment screw. Teacher, please see that the students are writing the function down correctly. The adjustment screw allows the objectives to move closer or further away from the object. The stages of some microscopes are restricted from being raised so high that the low-power objective touches the slide. If you can't raise it further using the coarse adjustment screw, don't apply force. In some types of microscopes, it is the body tube rather than the stage that is raised and lowered. You should also note which direction you need to turn the adjustment screw. Now, look into the microscope with your eye about 2 centimeters away from the eyepiece and slowly lower the stage using the coarse adjustment until the image comes into view. This part here is the eyepiece. You will see that the eyepiece has a label saying 10 times, which tells us that the eyepiece has a magnification power of 10 times, or it magnifies the object 10 times. A common mistake people make when they use a microscope for the first time is to place their eye right on the eyepiece. If you do this, instead of seeing anything on the slide, you'll probably only see a reflection of your eye and eyelashes. Remember, keep both eyes open while looking through the eyepiece, as this will prevent your eyes from becoming strained. If the image viewed is blurred, you can use the fine adjustment to focus the image until it becomes clear. This is the fine adjustment screw here. It is used to make small movements to the stage and helps focus the slide. The image is focused at the point where we have the clearest or sharpest image. If you find that the image is out of the field of vision, keep looking through the eyepiece and then move the slide very gently until the image is in the center of field of vision. In some cases, you may find that there is too much or too little light to view the image clearly. You can then use the diaphragm to regulate the amount of light that passes through the object. This is the diaphragm, and what it does is increase or decrease the amount of light reaching the slide. Now, write down the function of the diaphragm as it appears on the screen. I will give you about 1 minute to write down the function. Teacher, please could you make sure the students write down the function correctly? The diaphragm increases or decreases the amount of light reaching the slide. Once your object is in focus under the low power objective and you want to enlarge the image, you can use the middle power objective. Hold any two of the objectives between the thumb and the forefinger and rotate the nosepiece until the middle power objective clicks into position. Now you can focus with the fine adjustment until you get a clear image. The reason we use the fine adjustment only and not the coarse adjustment screw is because the coarse adjustment brings about large movements of the stage, which can push the objective onto the slide. This can damage the slide and the lenses of the objective. Also, large movements of the stage will cause the image to become unfocused, and it will then be difficult to refocus the image. Now that you have enlarged the image using the middle power objective, you may need to increase the diaphragm opening if more light is needed. Once your object is already in focus under the middle power, you can focus it using the high power objective. Taking hold of any two objectives, rotate the nosepiece until the high power objective clicks into position. Take care that the objectives do not hit against the stage clips when moving them into position. Now you can focus with the fine adjustment until you get a clear image. Remember that you should never use the coarse adjustment when focusing with the high power objective. You should not try and focus on an objective directly with the high power objective or with the oil immersion objective. First, locate and focus on the object with the low power objective, then shift to the medium power and then to the high power magnification. Here are some important points to remember in focusing. Firstly, never focus upwards or downwards without looking at the objectives from the side of your microscope in case your objectives are too close to the slide. Remember, with the high power objective or an oil immersion objective, if your microscope has one, you should never focus using the coarse adjustment. Always use the fine adjustment, otherwise you may break the slide and possibly damage the lenses of the objectives. In this microscope, the object is magnified firstly by the objective and then by the eyepiece. So, if we want to calculate the total magnification of the microscope, we must multiply the magnifying power of the eyepiece by that of the objective used. The total magnification is equal to the magnification of the eyepiece multiplied by the magnification of the objective. This is equal to 10 times 4. So, the total magnification is 40 times. Now that you have some idea of how to use the microscope, we are going to show you how to make a slide showing the onion epidermis so that you become familiar with the plant cell and their structures. An onion bulb is made up of several layers of modified leaves. Each leaf has a delicate epidermal layer surrounding it, which can easily be peeled off. This epidermal layer consists of cells that can be easily seen under the microscope. You will need the following materials or apparatus to make the slide: a small beaker with water, a dissecting needle, a pair of forceps which we use to pick up small objects, a scalpel which is used to cut our specimen, slides on which we place the specimen or object to be studied, cover slips which are these tiny pieces of glass that we place over the specimen, iodine solution, droppers, onion bulb, and a microscope. First, we place a few drops of water onto the middle of the slide using the dropper. Then, we carefully peel the inner epidermis from one of the leaves of the onion using the forceps and trim it using the scalpel. The piece of onion epidermis is then placed into the drop of water on the glass slide, and the slide is covered with a cover slip as follows. Keep the cover slip at an angle and place one edge onto the side of the water droplet on the slide. The water will spread along the edge of the slide. Now, using a desiccing needle, the cover slip is gently lowered onto the specimen, making sure no air bubbles are trapped between the cover slip and the slide. Once it is in position, examine the epidermal cells that you have mounted, first using the low power and then the middle power objective. Notice how you are not able to see the parts of the cells properly. Why do you think this happens? Think back to the previous lesson for your answer, and I will give you a moment to write down your answer. Teacher, please see that the students are answering the question. The reason the specimen can't be seen clearly is because there isn't enough contrast between the different parts. Therefore, we need to stain the specimen. The different parts of the cell will absorb the stain differently, or there may be a chemical reaction between the stain and parts of the cell. As a result, those different parts will be colored differently, allowing us to see them more clearly. A number of dyes are used to stain the structure of cells, but for the purpose of showing plant cells, we will use iodine. To stain the slide, you need to remove it from the stage and place a drop of iodine solution at one end of the cover slip. Now, keep a piece of absorbent paper at the opposite end to draw the stain across the slide. Then, after about 5 minutes, the cells will become stained and easier to see. You are now ready to examine the slide under the low power and then under the middle power objective. Notice how the nucleus and cell walls are deeply stained. This is because iodine is highly absorbed by the nuclear material and cell walls. Also, there is a chemical reaction between the nuclear material, the cell walls, and the dye. We will be studying plant cells more closely in future lessons, but for now, you need to take note of these shapes of these epidermal cells. If you look at the cell carefully, you'll see that a semi-rigid covering surrounds each cell. This covering is called a cell wall. This is important, so I'll give you a chance to write down. Just below the cell wall, you should see the cell membrane. And below the cell membrane is cytoplasm. While this oval or round structure is the nucleus. Now, it's time for you to do some work. I want you to draw 2 or more adjacent cells and label the parts that can be seen. I will give you about 5 minutes to complete your drawing. Remember that all drawing should be done in pencil, using clear lines to represent the different parts. When doing a scientific drawing, the idea is to represent the different parts clearly, not to make an artistic impression of what you see. So, please do not shade the drawing. Also, when you label the parts, firstly, make sure that your label lines do not cross. Then, write the label next to the line. Teacher, please see that all students draw the diagram neatly. Well, I am sure you all enjoyed that exercise. Now, it is time to look at an animal cell. We will study the animal cell in more detail in future lessons, but now I want to introduce you to animal cells and the structures by making a slide of cheek epithelial cells. You will need the following materials or apparatus to make the slide: a microscope, glass slides, cover slips, a dropper, methylene blue, a toothpick, and a beaker with water. First, place a few drops of water onto a clean glass slide using a dropper. Now, gently scrape the inside of your cheek or the inside of your lip with the broad end of a clean toothpick. Make sure you do not dig too deeply or you will cut yourself. Also, take care not to just pick up the white substance between the teeth, called plaque. Use the toothpick to transfer the scraping into the drop of water on the glass slide. Now, place a cover slip on the specimen as we described when making the onion slide. We can now examine the slide under the low power and then the middle power objective. Are the parts of the cells clearly visible? Again, as with the onion epidermis, you will not be able to see all the parts clearly since the slide has not been stained. To stain the slide, this time we must remove the cover slip and add a drop of methylene blue. Cover with the cover slip and again wait for about 5 minutes for the cells to become stained. Now, examine your preparation, firstly using the low power objective and then using the middle power objective. You can also carefully use the high power. You will notice that the animal cell does not have a cell wall. Instead, it is coated in a cell membrane. You can clearly see the cell membrane here. It is important to remember that one difference between plant cells and animal cells is that plant cells have cell walls, while animal cells do not have cell walls. This is an important point, so I suggest you write it down. Notice how the nucleus of an animal cell is located in the center, while the nucleus of a plant cell is not. The reason for this is because plants need more water. Therefore, plant cells have larger vacuoles at the center of the cell. This means the other organelles, including the nucleus, have to occupy the outer area of the cell. Can you now distinguish the nucleus and the cytoplasm? Yes, you can see the nucleus here and the cytoplasm. Well, I am sure you all enjoyed the drawing in this lesson. And by now, you should all know how to use the microscope and how to make a slide. You should also have an idea of what plant and animal cells look like. Thank you for joining me. Goodbye.